Hello and welcome to the Biology Chapter 12 DNA Replication Notes. This is Section 3 in the textbook. So this is an assignment for you to complete these notes, and I am going to go over this in a recorded format so that you can listen to it at your own speed, pause where necessary, and fill in the blanks as you see in the PDF. These are student notes. They're graded. We're 10 points, and you need to fill this information in. If you would like more information, obviously this is from our textbook chapter 12 section 3, so open up the book and you'll see some additional diagrams and good explanation for the pictures and the process, because this is how DNA makes a copy of itself. So copying the code. In a previous class we talked about how DNA has 4 bases, the A, T, C, and G. And these are strung together in 10,000, 20,000 nucleotides long. If a cell is going to make a copy of itself, we know that the DNA has to be copied as well. Recall that DNA is what chromosomes are made up of. So if we have 46 chromosomes, then all 46 chromosomes have to be made into a copy during interphase and the S phase of interphase. And DNA is double-stranded, so each strand of the double helix has all the information needed to reconstruct the other half by the mechanism of base pairing. And these strands are said to be complementary. And what that means is that C pairs with G and A pairs with T. So fill in the blanks there. Moving on. DNA copies itself, and this is the process called replication. The DNA molecule is going to pull apart in the middle, as you can see from the diagram. And then each single strand is going to make a new strand. So you can see from the picture that it is labeled original strand, and then it also is making a new strand, appropriately named new strand. It is described as unzipping because it does look like it is a closed zipper with 2 sides, and then we've all had a zipper kind of break and open up from the middle of the zipper instead of from the end where the attachment is and where it's supposed to unzip. So doesn't it look like that in the picture? By unzipping, it allows both of the sides to make a copy. And you can also see in the picture that each side of the DNA has an old copy and a new copy. So when it unzips, it has what is called a replication fork, and that is labeled in the picture on both sides where it is coming apart. That just means that is where the replication is going to take place, right there as it unzips. And each new strand is going to add new bases following the rules of base pairing. So A is going to pair with T and C is going to pair with G. And that means adenine is going to pair with thymine and guanine is going to pair with cytosine. Each DNA molecule is going to have an original piece to it and a new piece to it. And so fill in the blanks there on that sentence. It's important for you to understand that you don't get, like, the top piece is not all brand new. It has an old strand to it, and it made a complementary base pair. So now it has a new strand along with the old strand. And the same thing with the other piece of DNA at the bottom of the picture. Our chemical reactions do not happen quickly. It really requires enzymes, which are a special type of protein, and they speed up the chemical reaction. So this process of making a copy of DNA can happen very quickly with the help of enzymes. So there's an enzyme that is going to unzip the molecule. You recall when we talked about the structure of DNA, that the two sides are held together by hydrogen bonds. Hydrogens are always involved in this process, so that's where the name comes from. But hydrogen is slightly positive because that's where its proton is located, on the outside of that molecule, and it will be attracted to a negative side of another molecule. So there's an enzyme that is going to really push those two sides apart, and that is what the replication fork is. And then each strand is going to serve as a template for making a new strand. Now, once it's unzipped, we have to start making the complementary base pairs. And the enzyme responsible for doing that, it is called DNA polymerase. Enzymes are always named for the molecules that they're going to work with. So making a copy of DNA, so it carries the name DNA. And polymer is in the name polymerase. So DNA is a polymerase, which is a large molecule made by small building blocks or monomers. Keep in mind that all enzymes are going to end in the last three letters ASE. So just by looking at DNA polymerase, you should know that that means that it's an enzyme acting on DNA, polymerizing it, which means making it. So this enzyme is joining the individual nucleotides. If there's an A, it is going to bring in a T. If there's a C, it's going to bring in a G. Another cool thing about DNA polymerase is that it proofreads, something that all of us have a little difficulty doing, right? You write a report and you just type it out and then turn it in without looking at it. But autocorrect may have changed a typo that you had and changed the entire word. Now, DNA polymerase can also proofread that it has done things right. So occasionally it will erroneously bring in a C when it should have brought in a T, and it will catch that. It's interesting that our chromosomes have an ending, kind of like a cap. If you think of the aglet on your shoelace, that's that little plastic cap. It's there on your shoelace to try to keep things from unraveling. And there is a tip of a chromosome known as a telomeres. And these are the ends of DNA molecules to protect the chromosome. And over time, DNA may actually be lost from telomeres each time the chromosome is replicated, and so the telomere keeps getting shorter and shorter. Researchers believe that this is the limitation of how many times a cell can make a copy of itself when the telomeres are no longer long enough or doing their job of protecting the chromosome. The cells of prokaryotes and eukaryotes replicate their DNA differently. So we'll talk about the prokaryotes. Prokaryotes have a single, it's a loop actually, but it's just one strand of DNA. And it's quite simple. It's so simple that they're able to do a lot of research with prokaryotes, and we're talking about bacteria here. Bacteria are prokaryotes. Eukaryotes, on the other hand, like all cells that have a nucleus, that's a eukaryote, they have dozens or hundreds of places on the DNA molecule that get replicated all at the same time. With prokaryotes, there's just one replication fork that zips all the way around, but I'll show you a picture where the eukaryote may have multiple places where it starts to unzip. So there's the picture of the prokaryote replication, and these are bacteria. A single point opens up, and you can see in the picture where it shows the origin of replication up there in the top right, and then it's going to unzip in both directions until they meet on the bottom left. So prokaryote DNA replication produces a copy of the DNA, and the replications are attached to different points inside the cell membrane. So this single loop of DNA is attached to the side of the cell wall. So when the cell splits in two, then the DNA is attached to its appropriate side so that each new cell gets a copy of the DNA. So here is eukaryotic DNA replication showing two replication forks, but keep in mind there could be dozens or, as I mentioned, hundreds of these. And then the replication forks will join each other. Our DNA, for example, with humans, is on 46 chromosomes, and we could have 30,000, 50,000 or more nucleotides. So it would take too long if it opened up at one point and then had to just zip down that line. So it opens up many replication forks. So we're talking about how DNA makes a copy of itself in interphase and the S phase of interphase. But our next unit is on how do genes work. Now imagine if this picture was one gene. Genes, as we're going to see in our next unit, are code or a recipe for making proteins. So this has to open up just like this, and a copy of it is made, and that copy is messenger RNA. So the replication process is very similar to the beginning of making a protein. The two copies of DNA are produced by replication in each chromosome, and now you have two copies. These are the sister chromatid copies, and then the cell's going to enter the mitotic phases of prophase, metaphase, anaphase, telophase, and stuff. And in order for mitosis to occur, these two sister chromatids that were long, fragile pieces of DNA that are going to condense into tightly packed and wound up DNA, then it's called a chromosome. And that's when it's visible inside the cell. So that's the story of how our cells make a copy of DNA.